Okay, so the idea behind this is to go through the whole of one of the flow cytometry screens that we've been doing and go in a bit more detail about some of the technical aspects that need to be considered. I've not done this tutorial before, so we'll see how it goes. Yeah, feedback at the end, I guess. Okay, so you saw this, this slide from the talk, basically explaining the two types of screens, the survival screen and the sort screen. So it's going to be the sort screen that I'm going to focus on. In the cloud folders, I've put a number of files. Some of those will be for later. We're going to try and do some of the analysis. Initially, I'll go through the sort of technical aspects. So at least in theory, you'll know how to do one of the screens. Once you actually get into the lab and start splitting cells and things, that's where it all goes wrong. But yeah, we'll go through the theory, and then I'll try and take us through some of the analysis, which actually tends to be a bit of the bottleneck when people are looking to do these kind of projects, because a lot of the time it's molecular biologists doing this and get all the way to the analysis, and then the analysis is the bit that's kind of a bit trickier. So we'll try and go through that and make that a bit easier. Yeah, okay, so, and the other, I should mention, the other things that I've put into the folder, as well as some of the files that will let us have a go at doing some of the sort of analysis. There's a folder in there called protocols. And that's basically got all the protocols that we pretty much use and written up. I don't know how beautifully they've been done, but they give you an idea. And there's kind of individual protocols for some of the different steps that you go through. So that should be a kind of useful resource. Okay, so to start off with, and this is kind of the steps that we tend to go through with new collaborators when we first kind of set up a collaboration to do a screen and they've not done one before. It's tackling each of the hurdles one at a time. And the first thing that we actually have to look at when you're setting up the screens, the first step, you're looking to transduce your cells with your library. The question is, okay, how big does that have to be? How many cells do you need to actually transduce? And this can often be a bit of a block in doing certain things, because as we spoke about earlier, for example, if you want to do it in primary cells, it can be tricky to do a genome-wide screen because you need a lot more cells. You want to just work out exactly how many you need. Then it's a question how many you want to sort. So that's the next step in these kind of more tricky sorting screens. How many do I need to sort into the populations? And it's all to do with getting statistical power. So the answer, how many, is always as many as you can, but there's a practical kind of payoff between the two. You know, you can't be sorting for 7 days. Maybe you can at the Broad Institute, but most places you can't be sorting for 7 days, and you know, your cell's not going to be happy after that time anyway. Then the question we'll look at is how much DNA should we be getting from our cells? You know, that's another point tends to where things might go wrong. You know, you've sorted enough cells, you're like, great, and then you do the DNA extraction, you get a lot less DNA than you were expecting, and that's kind of like, well, that's going to massively reduce the power of the screen. Then I'll talk a bit about how we actually make the libraries for doing the sequencing. Again, the quality of your libraries will determine how many sequences you get back. The more sequences you get back that you can map to your library, the more power you have in the screen. So that's another key kind of step, getting that we had to do a lot of optimization to get those Illumina libraries really nice, that you get really good sequencing data. Okay, so the first step, how many cells do we need to transduce? To calculate this, we need to know a little bit of information about the libraries that we're using. I'm giving an example here of the chicken library that we're using. If you've got your own libraries, then the calculations are going to be different, and they depend on how many guides you have in that library. The libraries we've been using is based on this plasmid that's available at Gene, and the URL for that is just there if anyone's like looking to get the plasmid itself. It's called PKLV2, and essentially the features that it has is it's BFP, which is quite handy, and I'll show you later why. It's also got puromycin, which is essential because you need to do a selection step. Then it's got a U6 promoter driving the guide sequence itself. And then the rest of it is basically just common components of a lentivirus backbone and the plasmid for maintaining it in the bacteria. So for this library, we based the design on Gallus 6, which at the time was the most recent assembly available for chicken. Your library is only going to be as good as the annotation of the genome that you're working with. And for a lot of the animals that we're working in, that's obviously a problem because the annotation doesn't seem to be as good as, say, human. So for example, if you've got a gene and you're designing 4 guides based on the annotation, that annotation's wrong, and those guides might not work. So it's a real sort of issue. I think the annotation of things like pigs and chickens, they're getting a lot better now, so it's less of an issue. It is an issue for is the TRPC CRISPR activation screen I was mentioning, because for activation, you need to target the promoter region just upstream of the transcriptional start site. You've got a 300 base pair window that you have to hit. So you've not got a lot of sequence to work with. If the transcriptional start site of that gene is wrong, which in chickens, pigs, and things like this is often the case, then you're going to miss and none of your guides are going to work. But for the knockout ones, you've at least got the open reading frame to go for. Okay, so for our library, we had 3 to 4 guides per gene, targeting around about 18,000 genes. That worked out as, I've forgotten how many targeting guides there were, but we also had 529 non-targeting genes, so a total of 71,000 guides for this library. Not a massive library, but it worked for us. Okay, so that information is going to be key, figuring out how many cells you need to transduce. And what you're looking to do is avoid skewing of the library. What this means is if you've got 71,000 guides, you want to have representation of all those guides. You want to have them at a level that is going to be statistically significant. So if you're doing a screen and you've only got 1 cell with 1 guide, that's not going to really tell you much. You need to have multiple cells in there so that you've got statistical power. So what they kind of suggest is a minimum of 300 guides coverage. That means 300 of each single guide. Yep. And again, it's this question of 300 is the minimum. If you go for more than that, that's better. So you want to give yourself a little bit of leeway. The other aspect of doing these screens is, for the most part, you really want to have a single guide per cell. So you want to have each cell having a single knockout. You don't want to have cells that have multiple guides in there, because what will happen is when you do your sequencing, that cell will give you a certain phenotype. So for example, if it's low virus replication, that'll be that cell has that phenotype. It will count every single guide in that cell. So if only one of those guides is actually giving you the phenotype, you're also counting the other guides, and those other guides will add to the noise in the background you have in that screen. You essentially want to have 1 guide per cell. To do this with the lentivirus transduction, what we do is we transduce at a low multiplicity, so a multiplicity of about 0.13. That kind of ensures that almost all of your transduction events are single cell event. Yep. You're starting out with more cells than you actually need for your library coverage. Make sure that you're getting that just single cell transduction. Then because the lentivirus has puromycin, you can then select out any of the cells that haven't been transduced. I'm using the puromycin selection, that gets rid of those cells. And it doesn't affect your coverage because those cells don't actually have a guide in them. Yeah. So the take-home message from this is you obviously need more cells to be able to cover all your library. Okay, that gets us to the first question, which is how many cells do we need to transduce? I just realized so some of you are kind of struggling to get the access to the cloud and the presentation. Essentially, what you need is the information from these last 2 slides to calculate how many. This slide, which tells you how many guides are in the library. If you note that down, So 71,035 guides. Then take into consideration the coverage that you want. We're just going to say 300-fold, and then multiplicity of transduction. Okay? Who can calculate how many cells we need for this transduction?
6.5.
Correct. No tonics tea cake for you. Allowed to use calculators. You're not allowed because you do screens. Okay. Everyone get ready for a flying tonics tea cake. Oh, that was a good throw. That's a slightly mushed tonics tea cake.
Okay.
Is it okay? All right. So yeah, around about 70 million. And the calculation is essentially you've got 20,000 guides. Yep. You need the coverage that you're aiming for, say 300. So you multiply that 70,000 by 300. Yep. Then you've got to increase it by the number of cells that you're transducing to make sure you have a multiplicity of 0.3. So you take that and you divide it by 0.3. Yep. That increases it up to the. So what that will mean is you've got 71 million cells, and only a third of those are going to get transduced, and that gives you the. Mental arithmetic, terrible. Yeah, that gives you the 21 million. You'll have 21 million cells with the lentivirus in it. And that'll give you 300-fold coverage. Hopefully that kind of makes sense. Then essentially you've got to adjust that. So we were talking about the guys trying to do the salmon stuff. Because of the multiple alleles created by the genome duplications, you have to have much bigger libraries. If you have a bigger library, it means you've got to transduce more cells. So practically speaking, I think when we do this, we're kind of doing about 10 to 15 T175 flasks of cells when you do the initial transduction. So this is where you need a cell line that replicates fairly well. If you've got cells that will only replicate a number of divisions or replicate slowly, then it's going to be a pain. I should say, if anyone's got any questions, then just shout out. Question. Yeah.
How are you sure that you're getting 300 copies of each guide?
That's a good question. So that is the perfect scenario which doesn't exist in molecular biology. When you make your libraries, and I haven't gone into making the libraries because that's a bit more complicated, and we leave that to a guy who's a bioinformatician, and has those skills. When you make your library, it's the same question. You want to maintain coverage of all the guides as much as possible. So companies make the libraries and then they check them by sequencing, or you can make them yourself by cloning, shotgun cloning. One of the key steps is making sure that as much as possible, you have equal coverage of all the guides. In a perfect library, you would have 300 copies of each guide. That isn't the case. What you see is a kind of S shape. You'll see it later on in some of the sequencing analysis we do, where some of the guides just get lost from the library. And you have to just live with that because you're never going to get everything. Other guides are going to be represented at 10 copies. Other guides are going to be represented at 400 to 500 copies. That's why it's sometimes good to go above the 300-fold. If you go to 500-fold, you're going to get more coverage of all the guides. Any guide that was at like 10 with 300-fold will be a bit higher. If you go lower, where you start losing cells or losing the power of your screen, your coverage starts shrinking because the ones that were at low number now go to 0. And when you transduce your cells, that lentivirus is just not in there. So you're no longer going to get results from targeting that gene. You're going to lose that coverage. And I guess when it comes to it, whenever you do these large-scale screens, you're not saying that you're going to identify every single gene that's important to a virus. You're going to say we're going to identify some of them. We're going to live with the fact that we're going to lose a bunch because that's just the nature of large-scale screens. Yeah. Okay, so that's the first question. You've got to take into consideration how many cells you're going to have. It will also depend on the efficiency of your transduction. So some cells transduce much better than others. What helps a lot for that kind of thing is the lentivirus that I've said that we use, the PKLV2, has BFP fluorescence in it, and that was really quite helpful. This is just a slide that shows sort of optimization step where, as I said, what you're looking for is a multiplicity of 0.3. So you're looking for about a third of your cells to get transduced. And that's going to be more efficient in human cells, for example, because what we're using is a primate-specific lentivirus, so it works better in the cells that it's supposed to transduce. Less efficient in pig cells, then less efficient again in chicken cells. So you have to do an initial step where you're defining how well your lentivirus goes into the cells and how much of that lentivirus you need to put into your cells to get 0.3 multiplicity. And so this just shows transductions. You've got untransduced cells here, and you've got GFP here. We've got GFP here, but we're not actually using GFP in this. Untransduced, you've got no fluorescence. And then you've got 1 in 10, 1 in 20, 1 in 30, 1 in 40, 1 in 50. This is just the dilutions of the lentivirus. We haven't used actual titers. We're just going with dilution. And you can see at 1 in 10, you're getting over 60% of your cells transduced. And so you don't really want that because you're going to have the scenario where some of the cells are getting multiple lentiviruses, and that increases the noise. You want to dilute that down. And what we're getting here is the perfect of dilution is about 1 in 40. That gives us 33% transduction. We can see going from no BFP, the cells having BFP up here. Yep. So then you essentially scale that up. So you've got your 10 to 15 flasks, and you're hoping that lentivirus that you've produced is of good enough titer, you can now transduce that many flasks at that multiplicity. Then it's quite nice as well. After this, I don't actually have pictures of it, but once you transduce your cells, you're then looking to select those with puromycin, get rid of all the untransfected cells. And at the end of your selection, which we tend to do for about 10 days, splitting the cells as you go, you can then do this experiment again. And what you should see is pretty much 100% of your cells are now BFP positive. And that just shows that your selection has also worked. And this just, again, shows what's going to happen when you go from your transduced to your selection process. Kind of see it, not very well. Essentially, what you tend to see is you do see a lot of cell death once you put the puromycin on, because you're killing 2/3 of the cells, and the puromycin works pretty quickly. We tend to also split the cells as you go. It'll depend on your cells and how quickly they divide. But for the most part, we tend to split them once or twice during that 10-day selection period. Now, as you're selecting and the cells are growing, this shouldn't be a massive issue because the guides are now integrated into the genome. And so you can do a thing where, as long as you started with a coverage of about 300-fold, once you've done your selection, you can kind of grow those cells up to the level where you've got 500-fold or greater. You just keep growing those cells. You are going to get a bit of skewing of the library. And one thing you're going to see, as Victor was pointing out, is you're going to get loss of guides that are targeting essential genes. They're going to be lost, but you kind of want that because you don't want those in the screen. There will also potentially be guides that give cells a growth disadvantage or advantage. And they're, of course, going to grow faster and going to be represented more in the library. Okay. So we then get to the stage where we're looking to actually do the flow cytometry on your cells. So you've transduced your cells, you've selected the cells that you've got rid of the non-transduced cells with the puromycin, and then you do the flow cytometry. And these kind of assays, there tends to be a lot of optimization at that stage. I'm not going into it here because it tends to be very specific for whatever screen you're doing. For example, with influenza virus, we had to optimize the staining with the HA or the M2 antibody. And a lot of that was to do with which virus we used, because some of the antibodies worked well with some of the viruses like PR8. Some of the avian or swine-specific viruses didn't work very well. You've got to do a lot of optimization for this step, because the selection when you're doing your screen is critical. You really want to have nicely defined population of cells with a low and a high distribution. And if, for example, you have a virus that doesn't infect cells at 100%, you're going to have an issue where low population here, you're going to have a large tail of cells that aren't stained. Now, that's not because the virus is replicated poorly in it, it's because they've just not been infected at all. But they're going to carry guides, they're going to go into that population, and they're going to get counted. So that's, again, going to cause kind of noise in the system. You want to get this aspect of it really well kind of optimized. So, for example, we've got screens where we've done influenza virus, or IBV, or IBDV. Then we've got screens where it was like with the xenotransplantation, and each of them are really quite different, and you have to do that optimization to get it to really work well. Okay. Then I think for this screen, you can't kind of see it because the top's cut off, but essentially we started out with 130 million cells rather than the 70 million we talked about, and that's because when we do these sorting screens, we know we lose some cells at each stage. So we wanted to really give ourselves as much kind of leeway as possible. We started with 130 million cells, and then you basically gate, and again, you've got a decision here to make on what percentage you gate those high and low populations on. It depends on what you're looking for. So for example, if you're looking for genes that give you a really robust phenotype, then you're going to go for a lower percentage. So for example, here we've gone for 5%. Where are we? 5%. If you went for the bottom 2%, then you're going to only get the genes that are really critical for virus replication. Or you can widen it, say 10%, and then you'll get more genes that are statistically relevant for viral infection, but you'll include genes that are lower confidence or have less of an impact on the phenotype. So like a twofold effect on virus replication compared to tenfold. You can also, I've only done low and high and medium here. But you can split that into multiple. You can do the bottom 5 and then the next 5.
I have just a question about how much time do you leave the virus in the cells? Because I guess depending on how much time you would be selecting for infection or replication. So if you leave the virus for 3 days instead of.
You mean rather than not the lentivirus, the actual virus, so influenza. So yeah, I mean that actually turned out to be a critical part of the optimization that we did. I think my student was initially infecting for 16 hours with influenza and fixing the cells and then staining for the antibody. Again, it's this kind of payoff because if you do it too early, you don't get very good antigen production. And so the differential range you get on your screen is not great. You just don't see very good staining. Leave the virus on too long with influenza, it starts killing the cells. What you end up with there is not only less cells, you end up with a lot of debris. And when you're sorting the cells, debris starts messing with your cell counts and things like this and can kind of stick to things. The debris also still has signal from the virus. So that was one of the key things. So we started at 16 hours and we found that we had to pull that back to 12 hours to really get a good kind of signal and balance between signal and cell death. And the student was delighted with the 12-hour time point. And, you know, having to, but, you know, these are the things you have to do when you're a student. So yeah, so that's kind of key. Also getting single cell suspensions is really important. Because if you have cells stuck to each other, you're going to have this situation again where you've got a cell that's stuck to one that has a different phenotype and it's carrying a guide. And the cell sorting machine, you know, you tell it to chuck out anything over a doublet. And if you've got lots of cell clumps and doublets, then they're all going to get chucked out and you're going to lose the number of cells. We had a number of experiments early on we would start with like 130 million cells, and then we'd sort them. And given it was supposed to be 5% of the population, we were calculating, okay, that's how many cells we should get. We would actually get a lot less than that because the sorter was chucking them out. What we found was key for that, for some of the cell types that really kind of aggregated, was doing the experiments with the cells at low density so that they hadn't formed really tight junctions. And that way you could kind of lift the cells off relatively easily, put them through filters and get them into single cell cultures. That was kind of a critical optimization step. Okay, so you then got your 130 million cells, you sorted them, and in this was an actual experiment we did, and we got kind of 8 million for the low and 7.7 million for the high. Yeah, which works out about right. So that's a key point, is just check that you've set it for 5%. When you've sorted through your cells, are you actually getting 5% of the cells? The first thing is you can just check what the machine tells you. But what we found was some of the first times we were doing this, we would sort the cells, it would tell us we had 8 million, then we would take it up to the lab, spin the cells down, resuspend them, and then manually count them, the hemocytometer, and we would have 200,000 cells, which was very depressing. So it's really important that you actually double check how many cells it says you're getting and then how many you're actually getting. And that can be to do with cells sticking to the side of tubes and things like this. But here we got pretty good recovery of the cells, about 5% in each case. So the next question I've got is how much DNA you should be getting when you harvest these cells. So in the protocol, I've put a protocol that we use. We use a kit for extracting the DNA from the cells that we get. The cells, we have to fix ourselves, because if we don't, the virus keeps replicating. We get, it takes hours to do the sorting, so that doesn't work. So we have to fix the cells. Getting DNA back from fixed cells can be not highly efficient. So in the protocols folder that I have on the cloud there's a protocol for extracting DNA from fixed cells. Works reasonably well for us. So if we think about it, we know that we get about 6 picograms of DNA from a mammalian cell. And then the chicken genome is about 40% the size of a mammalian cell. So how much DNA should be recovered? Again, there's a Tunix tea cake in this. If you've already won a Tunix tea cake, you're not allowed to answer. Do screens, you're not allowed to answer. Hoping someone like closer gets the answer right. I'm worried about smacking someone in the face with a Tunix tea cake. Just the answer for the low population if you want. Anyone got the answer? Is everyone being shy, or do they just not want to be pelted with a... 19... what is that in micrograms?
Yeah. 1,000 picograms is 1 nanogram. So, 90.
Micrograms?
Yeah.
Okay, you coming?
I'm not good at.
You'll be fine. Here we go. Again, I think the throw was good. You guys need to really work on your catching here. It's almost like you haven't caught Tunix tea cakes before. I don't know. Okay, so yeah, about 20 micrograms. So that's what you're sort of looking for optimally. What we got from this screen was quite a bit lower than that. And that comes down to, this is the theoretical. If everything was perfect, you would get 20 micrograms, but these cells are fixed. So the efficiency that you get back from that is a lot lower. So what we actually got was about 6.6 micrograms. The next question, and this is a bonus one, hard maths, definitely comes with a tonics tea cake. What is the equivalent coverage we're getting 6.6 micrograms? In that each genome should carry one guide. I haven't done the calculation. What did you get?
100.
Not what I get.
30 or 40.
Yep. Oh, 37 fold coverage. So essentially you do, what is it? It's 6.6 micrograms, 2.5 picograms per cell. You divide the 6.6 by the 2.5, and then you divide that by 70,000. Gives you about, 37. So that sounds quite low. It's quite, I had a bit of practice on this one, so. Got it? Okay, that's the first catch I think we've had. It was a bit of a fumble, but it's improving each one. Yeah, and so that kind of sounds quite low, but what you've got to remember is these have now been sorted into populations. This is like 5% of the entire coverage of the library you now have in that. So you don't expect to have 300-fold coverage. You expect to have less than that. I guess if you actually divide the 37 by 5%, it actually gives you quite high coverage, like 600-fold or something like that. So this is looking like quite a good powered experiment. You have the DNA. The next steps are to make your sequencing libraries. So you have your genome DNA, and you need to essentially extract the guide sequence from that genomic DNA, that into a library, sequence it, so you know how many of each of the guides are in those populations. That's the key information that we want to get from these screens. Okay, so there are different protocols that you can use for this. A lot of them are published. We used a two-step PCR process to generate Illumina libraries. Again, this is kind of an example because not everyone will use Illumina. Some people use different systems, and so you would have to adapt to whatever you're using. I'll go through what we pretty much did for our screens using the Illumina sequencing. The steps that we've got from doing the experiment to doing the actual analysis, generating the libraries with a two-step PCR, then got to send them for sequencing to your favorite sequencing facility, pay the money, and then you get your sequences back hopefully relatively quickly. The next step is to download that sequence data. You need to map, because what you get is essentially big FASTA files. It says, here is every sequence we've got in a massive big file. You want to take that, and you need to map that to your guide library. So it'll sort of say guide number 68 is represented 40 times, guide number 103 is represented 1,000 times, that sort of thing. So you need a script that basically takes the FASTA file, maps each of those reads to your specific guide, and then that's going to give you the guide counts, which you can then do the further analysis from. We do the further analysis using the guide counts using Magic, which is the broad tool that they developed, is online. It's available on Galaxy and is relatively easy. Pretty much anyone can do this step. Previous steps can get a little bit tricky, and usually, certainly we had the help from Nick Parkinson who did this informatics aspect. Then there's the data presentation. I'm not really going to go into that too much because it tends to be a less specialized thing and just depends on what program you're using, Excel or the other ones I've forgotten. the newer ones. GraphPad and these kind of things. And they, or if people are more adept, they can use R to generate various different things. So we'll just go over some of the basic kind of presentation. Okay, so the first part is doing the actual generation of the libraries. As I said, we use a two-step PCR system for this. The first step is the genomic DNA extraction. And as I said, there's a protocol in there and a kit that we use that works pretty well. Within that genomic DNA, for each of the genomes, you've got a single lentivirus that carries the guide RNA. The first step PCR is using PCR primers that recognize the non-variable regions on either side of the guide. So it's just recognizing regions in the lentivirus. Yep. That way you can amplify all guides pretty much the same. And that's going to give you a PCR product of about 260 base pairs. You've got one aspect that's kind of key, and it has this thing that's called a stagger in it. So your reverse primer is just a general primer that recognizes the bit in the lentivirus. The forward primer is actually a pool of 10 different primers. And it looks like, the actual sequence looks like this. So the bit in green is the bit that actually recognizes the lentivirus. The bit in black, we'll see later, is a landing pad for the Illumina primers. And then the blue bit here is the stagger sequence. And you can see it goes from having no nucleotide, having, I think it's 8 or 9 nucleotides at the bottom there. Okay? So you're doing your PCR, you've got your genomic DNA, you've got a reverse primer, and then you've got this pool of forward primers. So can anyone tell me what the stagger is for? Not people who do libraries. Definitely not people who do libraries. What is the reason for having this pool? It's quite important, and it's to do with the actual Illumina sequencing. It's not to do with anything to do with the library preparation. We've already got a mixed cake. Nope. I want to have a guess. It's to do with when you're doing the sequencing with Illumina, and you have essentially your stepwise process of you add a nucleotide and it's fluorescent, and the machine reads the fluorescence. If every sequence right at the start is the same sequence, the whole plate ends up with the same fluorescence, and it sends the machine off a bit. It doesn't like reading the same color all at the same time because you get massive bleed through. What you want to create is complexity in the initial steps of the sequencing. What this does is means you've got a pool where guide sequence may be all the same and the primers all the same where it's reading, but it starts reading at that point. And so at least 1 in 10 of them have an A or a G or so forth, and then along the way. So it's essentially to make the sequencing more efficient.
Yeah.
So I get that one. Yeah, so, and in the protocol, they've got that about the primers. It ends up, primer cost is actually a wee bit to take into consideration when you're doing these, because when you generate the different primers that are quite large, as well as your reverse and the second primer sets, it gets a bit spendy. Okay. Then the second step PCR, this is where essentially you're adding the standard Illumina primers, and they're just recognizing the sequence that we've incorporated onto the end of the first primers. Whoop, nearly. So in addition to adding the Illumina primers that allow for sequencing, there's another key aspect to this, which is the barcodes. So again, we use standard Illumina barcodes, which is the I5 index and I7 index. These are essentially just kind of random, but relatively random sequences. And it means what you can do is pull your samples for when you do your sequencing. So say from your screen, you going to have samples from when you initially transduce your cells, day 2 or so. You're going to have a sample where you've taken it after your selection, your pre-myosin selection, so about day 10 before you do your actual screen. Then you're going to have your low, median, and high populations. So for a single screen, you're going to have 5 samples. And say you do that in duplicate, you're going to have 10 samples. And you don't want to run all those samples individually on sequencing because you're going to have 400 million reads on them. And it's going to be really expensive. What you can do is you have a set of these I5 and I7 primers have different barcodes. You can mix and match them. And so in the end, you can do about 20 different combinations of these primers. So you can essentially produce 20 different libraries that you can then pool together and sequence on one go on the Illumina run. And then you're just calculating the how many sequences you actually need to generate the coverage again. All make sense? It's not making sense, then just shout out. Yes?
So the first PCR is just to amplify, so you have more of your sequence for the barcoding, for adding the barcoding to the.
Yeah, it's essentially to get rid of, you've got all this genomic DNA, 6.6 micrograms, and most of that's DNA that you're not interested in. So you're just wanting to create an amplicon you can then easily sequence and gives you the guide sequence. Questions get atomics as well. You ready?
Yeah.
Clean catch. Nice. Okay. And so these are examples of the first and second step PCRs. This, again, required a bit of optimization for us to get into the sort of routine of doing these. Kind of the key things are the amount of DNA that you include in the first PCR, because you want to use all the DNA you get, because that's all useful of material and information. You want to cover any guide sequences in there. Anyone who does PCR and genomic template knows you don't want to put like 6 micrograms into one PCR because it's not going to like it and it's going to kind of, be very inefficient. You end up splitting that into serial PCRs. So for example, if you've got 6 micrograms, we tend to do between 0.5 microgram and 6 micrograms, depending on how many PCRs the student wants to actually do that day, because you've got this and then you've got your other samples as well. So they quickly start to pile up. It would tend to be about 6 PCRs. The other thing is you don't want to over-amplify the sample because that can introduce PCR bias. That's where some guide sequences will just amplify more efficiently than other guide sequences. So you want to actually amplify it the minimum number of cycles that you have to be able to essentially get product that you can measure on a gel, for example. I think we do around about 24 cycles. If anything, you should probably reduce that a little to less than that. And you can do this by initially taking some DNA that you've extracted from a sample and do a PCR where you do, sample the PCR at 10 cycles, 15, 20, and then try and narrow it down. Kind of a key aspect of it, because if you get PCR skewing, then it throws off all your results. So there's a bit of a kind of a change in philosophy when you go to the second PCR, because in the first PCR, you wanted to use all the material that you've got because that's covering all the guides that you've essentially collected. Once you've amplified that to a reasonable level, then you've actually got huge numbers of copies of guide sequences in there. You don't want to actually go from taking your first PCR and putting all of that into your second PCR. We found that we had to reduce that by about 5 to 10 nanograms per template. And you kind of think, well, now we're just throwing away all this stuff that we've just generated from the whole experiment. But because you've already amplified that, you still kind of maintain coverage. If you work out the Avogadro's number and the number of copies that end up in your PCR reaction after the first one, it's pretty huge. And it's key because we found if you put too much of the first template into the second PCR reaction, you start getting secondary bands. That has a big impact on your downstream sequencing quality. So key thing for your second PCR is you want a really strong band and very few secondary bands. Yes?
Do you inactive PCR from the first to the second?
Yeah, so for the first PCR, we tried a number of different kits and things. I can't remember exactly. I think it's a bead kit that we ended up using. I think it's Cymogen. It's in the protocols folder. And yeah, that did have, you know, an impact. You want it really nice and pure. The second PCR, do gel extraction. That was a real headache because, as you probably know, gel extractions are pretty terrible. I think, again, we ended up with a bead system doing that. The QIAGEN ones were horrible. And we would get like, you know, 10, 20% recovery, and the quality was really poor. So yeah, that's also fairly key. Yeah, it's pretty depressing when you do the second step and you use, you know, a whole ton of your first PCR and you get a nice band for the second PCR, and then you gel purify it and you get nothing back. Okay, so you've made your Illumina libraries, you've pulled those together, and you've sent them off for sequencing, usually by telling your PI that it needs sequencing, and he gets in the car and drives, because that's what I do for my job, apparently. And then initially, you'll get a quality control data back, and often the first times we were doing this, we would get our sequencing hub to actually generate this before going ahead with the sequencing. And this is just bioanalyzer data. And essentially, you're looking at is here is each of the libraries that we've made after the two-step. You can see a bit of a smudgy sort of band up here, which kind of, this is the main band, this is the library. And then here you get a little bit of a peak for the background band. And in the next slide, I think, once you've done your sequencing, you then get the report back. So this table here, it's a wee bit small and complex looking. Essentially what it's showing is each of the individual samples you've taken. So we've got our R17 is just the experiment number. So there's two repeats here, R17 and R18. First top samples here are unaffected at day 4. So this is after we've transduced, and very soon after we've started the puromycin selection. And we've got day 7 and day 11, and 14 here. The reason we did quite a few, just to sort of see the progression of the selection of the guides, also for the dropout of the essential genes. And you can see the I7 and I5 sequence. They're the barcodes. You can see they're slightly different for each of them. Essentially, once you've pulled all the samples, they all get sequenced. And then afterwards, they can bioinformatically separate those sequences based on the barcodes into your original sample pools. Then this is the key part here, looking at how well your sequencing has worked. This was, I remember now, it's like 100 base pair, one-directional sequencing, and I think it was 400 million reads for this one. P2, I think it's a P2 cell, they call it, or something like that. But overall, it was 400 million reads, and we calculated what you want to do is how many samples you've put in there. Say you've got 5 samples or 10 samples, you're going to want more reads to make sure you get full coverage. What you can see here is the number of reads we've got back from each sample. So at the top there, we've got about 50,000 reads for most of them. Then there's just a couple down here have less reads. That tends to be going back to what I was talking about, getting the really good quality Illumina libraries. If you're getting secondary bands in your PCR, it tends to really reduce this overall number. Yep. And then secondarily to that, when you then map your reads to your library, you get another drop if your library sequences aren't of high quality. And a lot of the reads will not match to your library. So that kind of reduces the power and makes you sad. All right, so this bit, I was going to try and see if we could go through this individually and download the files and do the script to generate the guide counts. So this is the step where you've got your FASTA files, and then you run that through, we do it through a Python script. It essentially matches each of the reads to the individual guides in the library. So essentially what you divide the pipeline, the FASTA file, the library, the guide library, so that's like all 70,000 guides with their names attached. And the pipeline basically matches those to them, produces an Excel file, which then says, all right, here's your 70,000 guides. This is the copy number. It'll be, ranging between 0 to, a few thousand, essentially. The FASTA files were really pretty big. They're gigabytes kind of thing. And it didn't work for me downloading it from our OneDrive from my postdoc. So I was figuring it probably wouldn't work on the cloud, and the analysis would take a while. So yeah. That's a good question. I think we allow 1 to 2 mismatches, depending on the quality of the sequence. So Nick, when he runs this, or now the postdocs in the script, I think you can change those variables. And if you run it and pretty much everything matches to your library from, maybe 10% or something like that, you're kind of like, just leave it. If you're getting low levels of matching, then that's kind of a worry. Then at that point, you kind of maybe start introducing mismatches, allowing for mismatches, and it increases the number of reads. And you kind of take that into consideration when you're analyzing the data as to the confidence you've got in the hits.
You don't mention how you make the library, the single guide library.
Yeah.
Once you clone, so you order the pool of oligos, you re-amplify them, you clone them in this PLK.
Yeah, PLKV2.
But do you sequence?
Yeah, that's key.
How often do you see errors of the synthesis?
How often do you see a sort of skewing or a poor library? Depends who made the library. So we have had people make them in the lab. Actually, that works pretty well because they care. And so they'll do it, and it'll, you have to use very high electrocompetent bacteria, very nicely purified DNA, get the ligation to work really well, and then, plate them out on huge plates where you get loads and loads of colonies. If you do all those steps well, then you get really nice, what's called a skew score. So what you want is the lowest potential skew score, which means, it's closer to a straight line. I think the maximum kind of skew score you would want is anything above 10. You're starting to look at that library and not be particularly happy about that.
Then you buy from the oligo first.
Yeah. Initially, GeneWiz, and they were supposedly producing very nice libraries because we got them to sequence them what we thought was afterwards. And then we sequenced them as well because we're not very trusting. And the results didn't match. The skew ratio was a lot higher when we sequenced them. So when we went back to them and said, what's going on here? They were like, oh, no, we sequenced the oligo pool, not the library. I was like, well, that's pointless if you can't even get your oligo pool, right? So there was, we've switched now to Twist. I think they're a bit more expensive, but so far the libraries we've been getting are of a higher quality. But honestly, if the highest quality might be worth, because you can buy the oligos quite cheaply, oligo pools. Yeah, compared to if you actually pay for the whole library, they tack on quite a big charge for the cloning. And, if you do it yourself and you do it well, you're going to have, I think the libraries that have been made at Roslin have been higher quality than anything we've got from commercial ones. Yeah, so essentially you download your sequence. It can take a while because they're really big files. You unzip them, and then you match the guides to the sequencing to the guide library. In the cloud folder, I think there's a folder called sequence analysis. In there, there's the pipeline that Nick has developed, and it's run on Spider, which is one of the Python formats. and there's instructions in there as well how to download Spider. It tends to be with the analysis. It's not the pipelines and the tricky things. It's the really basic things. Like, I was coming to do this, and I was like, okay, like, yeah, and you just load this onto Spider, and I'm like, I don't know what Spider is, and I don't know where to download it from. So there's a point by point how to download Spider, and then on the slides, but it's also in the protocols how you do the next steps. Yeah, so this is just the part about downloading and unzipping the files. When you download them, it's a FastQGZ, and then which will tend to be a big file, but the unzipped one's even bigger. It tends to be, I think, around about 10 gigs, something like that. Yeah, so this is a small part of the script that's in there, and you can look at the whole script if you want. Key parts that my postdoc is pointing out here is the bits that they change when they do each experiment. Leave most of the script the same. In green here, you're just putting in the names of the files. And the reason you change this is because each time the file names are slightly different. And the key part is you have to maintain the file names exactly the same as they are named the actual file. And also you've got to avoid any, is it called non-normal characters or something like that? So no hyphens, so you've got to change those all out as well. And make sure you keep the syntax the same, so the format exactly the same. So you put in all your samples that you're interested in, and then you also have on here, that's your guide library. You have your guide library, and that's also in the folder as well. Then this is just the name of your output file, which you get as a text file, but you also generate as an Excel one that's easier to read, and I'll come to that. Okay, and this is just, if you're using exactly the same primers as we're using, this will work fine, but if you're using slightly different primers, then you put the sequence that's either side of the guide. It just tells the script where to actually read, essentially trimming out the guide sequence. For the most part, this is, if you can just, everything has to be in the same folder. So the script, sequencing files, your library, you've got them all in the same folder, and you load up Spider, and you just make these small changes, and you just hit run, and that should generate your Excel file that will go into the same folder. So that's that step. If you're doing slightly different sequencing with a different platform or whatever, you might need a little bit of help from a bioinformatician just to get past this stage. Okay. So one of the first things you can do, because now you've basically got your nice data, which tells you each of your samples of the 70,000 or so guides, exactly how many guides you've got. And you can kind of do a quick look at the kind of coverage that you've got. The whole thing we're talking about, do we have good coverage? So what you can look at is the uninfected samples, both before and after puromycin selection. What they should look like is they should look something like the original library. So what you're aiming for is the absolute, perfection would be 300 in a straight line. But what you tend to see is a bit of skewing. But you're hoping that that skewing is not too massive. This is kind of what it looks like for a reasonably good experiment. And this is the one we did, and you've got the A11, and then afterwards you've got the sorted population here. And so, as I said, here, essentially you'd be looking at about 100. If it was a straight line, it would be perfection. You've got this kind of skewing. And so down at this end, you can see you've lost actually about 70,000 guides. So 70,000 guides are no longer in the experiment. And that's going to reduce your coverage of your, say, 20,000 coding genes that you're looking in chicken. Any of those guides that are just not there, if they're an important gene that the virus would normally need, then you're not going to see it in the screen or in the results. This is still fairly reasonable. We have had results where, for example, we tried transducing our cells, selecting them, growing them up, and then freezing them down. We thought, well, we can do that once. We can freeze these all down, keep them in liquid nitrogen. Whenever we want to do a screen, just resuscitate them, do our screen. We harvested the cells after we resuscitated them, and we did this sequence analysis, and basically it looked a lot more like almost a straight S line here, meaning there was massive skewing, and that was because when you resuscitate cells from frozen down, and a lot of cell death, it just doesn't like it. You may be able to do it in some cells, may be able to optimize that protocol. We just went, right, we're not doing that again because, you know, the screen after that was completely useless because the skewing was so bad. Okay, and so then we get on to the magic analysis. So we've got the file, the Excel file that has all the copy numbers of your guides. What you want to do is figure out statistical analysis that will tell you, because you've got 4 guides in there, so you're not obviously going to look at all 70,000 and sort of eyeball it across and say, all right, which ones are enriched and which ones are not. What we're looking for is a pattern where we see, say you're looking for a host dependency factor for influenza. We're looking for guides that you see a higher number in the low population compared to the mid and the high population. Yeah. And this package, MAGIC, which was developed by the Broad Institute, basically do that. It looks at the guide counts in each of the sequences, each of the samples, or the 3 to 4 guides for each gene. Then it gives you a overall fold increase or enrichment for that one gene, and the p-value, which is going to be based on, do we see the same pattern for all 4 guides, or maybe 3 out of 4 guides, or just 2 out of the 4 guides. Okay? So I was going to try and do this where we all do it together. What that will require, it'll probably take the rest of the time and lunchtime, but I don't know if I'm going to end up going offline on this as far as sharing. You guys could go into the folder for the cloud, which actually I'm just going to do it on my computer and hope that you guys are doing the same thing. Okay, so do you guys have the cloud up? And there's a folder called Magic Analysis. And in there, there should be a text file called something like R17, R18. Yeah. You download that? Put it somewhere where you can find. And then what we need to do is put this back to I think we can still share. It should be all right because I'll do it on here and they'll be able to follow along. Essentially on here, you guys could go to that website. I suppose people online, it's the one at the bottom right-hand corner of the Magic Analysis slide. That should take you to the Galaxy website. Maybe an issue when like we all try and do the same analysis at the same time. It could go wrong. Let's see. Okay. You guys mostly got onto that website and downloaded the file? Okay. You want to just open that text file, but open it in Excel rather than as a text file. Okay. You guys seeing a column on the left with all the guide sequences followed by the gene name, and then a number of columns that have numbers ranging from 1 to... Yep. So essentially, what this is, if you look at the very top row, target ZZZ3, obviously in alphabetical. These are each of the samples from one sequencing, one CRISPR screen, we've got 2 repeats. R17 is one repeat, and R18 is the other one. And you've got the first column low, then med, and high. And right at the end, you've got the uninfected ones. So for example, you can look at the second sequence down, the ZZZ3, and you can see in the uninfected ones, it's fairly well represented. You've got 500 for the library, plasmid library, about 1,500 for the uninfected A10, and 800 for the other one. And essentially what we'd be looking for, any major skewing in the low versus the mid and the high. So in this one, it's not really seeing much going on. There's about 386 in the low. If it was a hit, that number would be in the thousands, and you would see much lower numbers in the mid, the high level. Okay. So if you go back to the Galaxy analysis, the Galaxy website, the next step is on the box there, the search tools. If you enter in there magic, and then the drop-down menu that comes up is one called Magic's Test. You guys found that? You just click on that. The next thing we need to do is just to upload the text file you guys downloaded from the cloud. And to do that, I click on saying single data set. Then you see next to it, there's 3 dots. Now. And then you hit upload down in the bottom left-hand corner. And then you go to local file. Oh, yeah. That'll work. Okay. Ah, right. You want to stop sharing, then reshare? Okay. If you guys have got that far, if you want to hit start, we'll see how long this takes. You have to wait until it'll go green on the initial box, but then you have to wait until it goes green. Wait until there.
Which page is it supposed to be in?
Let's see. Yeah, we're about there. Over here, it should appear, and you have to wait until it turns green. This is the bit that I'm wondering, because we're all doing it at the same time, it might take a little while. Anyone's file turned green? Anyone not got tonic tea cake? Anyone just want a tonic? Better. It'll start really flinging them there.
You want this one?
Okay, so green. I've got to remember the next steps. Where are we? Okay, so now you need to name the treated samples, and it has to be exactly how it is in the text file or the Excel file. So if you still have the Excel file open, what we're going to do, we're going to compare the low sample with the median sample, both replicates. The way to do this is basically the heading, the low sample, if you take that and copy and paste it into the treated sample box, and then put a comma, and then do the same for the R18 low sample, with no spaces between, so just a comma between the two of them. I think it's essentially column C, very top heading. So it should, if I explain that, it should be in this box here. Yeah. The two samples, and it should read something like R17 underscore PR8 underscore low, comma, no space, R18 underscore PR8 underscore low. And then in the control sample box here, do the same thing, but with the mid samples. If you just copy and paste those, then we select positive selection because you're looking for genes that are enriched. That's under the advanced options box. Here. In the advanced box, if you scroll down to sorting criteria, change that from negative selection to positive selection. Then up in the corner here, it just says run tool. And then we just have to wait until that, hopefully those two in green. Anyone have any questions in the meantime? It's basically on orange and there's boxes. I think that's a good sign.
So like briefly walk us through like how you actually generate your like guide library. Like, are you required to have like, you know, like, are you doing it automatically?
Do you mean actually making the library? Do you mean designing the guides, or do you mean actually cloning them?
Bioinformatics.
Yeah, okay. That again, I didn't go into because it does require a bit more complicated bioinformatics. Essentially, what you're doing is you're starting off with a transcript file that has all the, you basically want to download all the open reading frames from a genome. So you would go on Ensemble or something like that, go to the genome platform that you're interested in, and whichever release it is, which assembly it is. So Chicken 6, and what Nick would have done was generate a file that had all the open reading frame sequences on it. So no introns, no exons, no introns, none of the 3' ETR, 5' ETR, just the open reading frame. You then want to essentially use pretty much some of the tools that are already being designed, often by the Broad Institute. Victor was talking about some of those. And you're looking to design guides that, first of all, they've got to recognize a PAM sequence. Yep. So that's NGG. No, yeah, NGG.
Like for Cas9, not like.
Yeah, this, I mean, it'll be specific to the Cas. I mean, they all work in similar sort of ways, but for example, the activation needs to be the promoter region, whereas the knockout needs to be the exon you're targeting. And then again, you want to target close to the 5' end of the open reading frame, because what you're doing is essentially it's going to cutting until it introduces an indel. And then you're looking for that indel to put the gene out of frame. If it's further up, further closer to the 5' end, the less of the protein is going to be read correctly. And you'll also probably introduce a stop codon at some point. If it's at the 5' end, more likely to disrupt the gene function. Yeah.
For the because you mentioned like using one of the already available tools for like the guide design. Like I'm actually required to have like a guide design tool that actually supports your genome host interest. Because for example, like you need to manually design stuff for like trout or shrimp because none of the online platform actually have the genome, so you can't check for off targets or anything like that. You need to check it manually.
Yeah, you need to basically develop a pipeline that will do that. So that's kind of Nick's domain. That's where he'll actually develop a pipeline that will generate in the right format all the the genome information in one file, able to put that into his pipeline. Pipeline then designs guides based on sequence, proximity to the 5' end. It'll then give it a score based on the parameters that Broad have kind of developed to see if it's likely to work as a guide. And then the key thing is scoring the off-target effect. And that takes the really big computing power. That takes the longest time because it's to take each guide and try and match it any of the other open reading frames. That's the one, for example, they'll do that on Eddie Roslin to have the compute power to do that. All that is beyond me. This is mainly beyond me, but that's really beyond me. Yeah, that's where we get like Nick to help design the guides. So whenever we're doing a new library, we'll basically say, Nick, how much time do you have? And what cake do you currently like? Name and cake.
Yeah. I was wondering, because designing guides for like stuff like salmon is okay because you can just like collect the genome on the platform and it's gonna check for targets. But I was recently trying to design the guides for trout, and none of the platform had trout genome, so I did it manually. I was wondering, like, I mean, obviously you're developing with knowing your pages ahead.
Yeah.
For rainbow trout, there is a full list of all the guides pre-designed and pre-scored by using rules reset on a Broad Institute webpage. If you Google rule 3 set, there are, I think, 20 or 30 genomes that have been done so far by the Broad.
Yeah, I think for the most part, the guide libraries are going to become more and more available, where you can just select a species and go, genome-wide library. And I would imagine, I don't know if there's a tool yet where you can select a function, say, for example, interferon or something like that, and it'll just produce a library, whatever species you want. I can't imagine they're that far off. It wouldn't be that difficult to do. Yeah, I mean, it would, because that is a bit of a bottleneck at the moment. You have to have someone who can do that if it's not available. Okay. Okay, have those turned green yet? Oh yeah, okay. So there's a little eyeball icon next to the icons, and I think if you go to just the top one there, and that's go to the middle one with the eyeball icon. That's essentially your data there, and it's already sorted it into a list of the top hits. So I have to admit, this was from a proper screen for chicken influenza virus, but because it was funded by a company, we had to scramble the gene names. So don't go kind of investigating ITGA4 as an influenza dependency factor. Essentially, what this means is ITGA4 if this was like a real screen, then this is producing guides that when you target that gene, you're getting enrichment of those guides in the low population. It essentially means that that gene is required by influenza virus to replicate. And essentially that's what it's meaning with all these genes in this list. You can download it by just clicking on the box. And then if you scroll down, there's a little disk icon. We don't use those anymore. Then that'll download it as a text file, and you can just open that in Excel. You guys able to open that in Excel right away? I think you can open it into text file. Then if you save that, has anyone been able to download that and open it in Excel? For some reason I'm having difficulty doing it. Okay, so then it gives you a number of values. I think it should look something like this. Is that what it's looking like? Yeah. The key columns here is you've got one on the far left, which tells you the gene name. And then the ones that we're looking at is the positive values, because we're looking at the enrichment ones. And the two that you've got is the p-value, so the ones that are sort of 2.75 to the power of -7, and then the false discovery rate. Okay? And they all are basically telling you the statistical confidence that you can have in that hit. Obviously, the smaller the number, the more statistically significant that hit is. Yep. And then over on this side, you've got the log-fold change. That's what I was talking about. It's basically taking the 4 guides together, and it's saying how much they were enriched based on in the low population compared to the medium population. So if you were to open up the original text file with the guide counts, so I think that was, and open up the Excel file. If you search in the gene column there, and you look for ITGA4, this is one of the things you can do when you get the results. You can actually just do a quick eyeball of each of the guides, because in the guide counts, you've got the separate guides, all 4 separate guides for the genes. So have you got, have you found the ITGA4 in the guide count Excel file? Yeah. So in the first column, you can see the guides range from about 200 to 1600. Yep, is that right? Or, for example, for R17 PR8 low. And if you look at the column next to it, that's for the median population. And they're all considerably lower than the low population. This one's a bit of an odd one, actually. I don't, no, that's the other low, yeah. And then the high is the same. Essentially what this is showing is for all 4 guides, you've got a significant enrichment of that guide in the low population compared to the medium and the high. And the same pattern you see for the repeat in R18. You can actually go back, and if you're looking to see, because the magic analysis, what we found is not perfect. Sometimes it can score genes quite highly. You'll go back and you'll manually look at the guide counts. And when you look at them yourself, you're kind of like, I'm sort of less impressed with that. What you can actually do is graph each of the guides, the count numbers. You can look at them just by eyeball and say, all right, does that actually look like all 4 of them are giving the same kind of clear pattern? Sometimes, specifically, it'll be thrown off if you've got guide counts that are really quite low. For example, if you've got one in the low population that's 6 and the other 2 are 0, 0, that seems like a big enrichment, but it's not really. It's just that there's low guide count and things are going off. So It won't tend to read that if that's the case for all 4 guides, but say you've got 2 guides that look pretty good and 1 that's really low, it can really push that up the scoring, and you know, it's worth looking at those individually. And then this is basically the data set that you then work from to do all your kind of visualization of the score. And as I said, I'm not going to go into that too much because you guys can sort of map this out the way you want. A lot of the time, as I showed in the talk, we tend to do volcano plots. This is just one done with Excel, so it only has it in one direction. I think you need to use GraphPad to be able to do the 2-directional volcano plots. You could basically take the column for from the file that you downloaded from the magic analysis. You can take the column for R17 low, plot that against the column for R17 medium, and it should give a volcano plot kind of like this. Again, because it's Excel, it won't label them specifically, but you can kind of work out from the values and label them yourself. And you should end up with a graph kind of like this. You see most of these are the irrelevant ones, and then you've got the ones up here that are statistically significant. They're the sort of targets that you want to go after. If you get a really good screen at high power, what you tend to see is all these start lining up and plateauing because you can only get a certain level of p-value, and it plateaus. If you get kind of 30 or 50 genes all like that, then you know you've got a really good screen. These kind of ones may be involved in things, but the confidence is a lot lower. Obviously, the more repeat screens you do, the better the power you get, more confidence you're going to have in the hits. Okay, that's us, and so I guess we're finished a wee bit early. 2 minutes early?
Yeah.
Oh, I thought I was supposed to go all the way to like 12 or something. Oh, yeah, that's true. I'm in UK time. That'll explain that. Anyway, are there any questions? One thing I should point out is, you know, we have all these reagents and we're happy to share them, and we have the protocols are now on that folder. And we're also happy to sort of, if people are looking to set up screens, we can kind of go through them as people are doing them. We're happy to sort of have meetings and just guide people through it. Apart from that, yeah, any other questions? Yes.
Talked about diseases and mentioned xenophobia. Do you have any other suggestions for phenotype things?
Yeah.
Yeah.
Well, we're doing parasites now, and we're doing bacteria. A lot of the time, it has to be something that's intracellular. So we're looking at those. The most part now, we're doing a lot of different species. So someone after, in pigs specifically, I think the genome-wide screens are nice. They do tend to pick out specific things that are heavily biased towards. So entry factors, anything that gives a really big phenotype. I think things that what I would like to do next is be more functionally specific, maybe smaller libraries. And if you've got it all depends on the readout. If you have a readout that works for high throughput, that's really what you need. So some of the things we're looking at doing is, say, treating cells with interferon, then doing the viral infection. Then you're adding an extra layer of complexity there. But if you can generate, say, a fluorescent readout for whatever you're interested in, then you can measure that.
We just don't know.
I mean, depends on your imagination, really. What screens you can come up with and whether they're practical. I mean, you know, transplantation, for example, I never would have thought of. It was those guys through the ASFE work, you know, they were like, oh, you know, we make these pigs with the knockouts. Also, we're interested in this and we think we can do a screen. And so, yeah, I mean.
The reference genome to which you are using to design the guides and the cell type in which, in principle, also from the same species, but you want to make the screen need to
Relevant.
if they're not
Yeah.
if they're not concordant, then you're missing a lot. And you're saying that you are counting how many guides are in the low, mid, or high. This is what gives you actually the final power. Depends on the alleles, on
Yeah.
what is the
If
system
you're
using.
if you're interested in milk production, then it wouldn't be very advisable to do a screen in skin fibroblasts. Yeah, because a lot of the genes that are important in milk production are probably not going to be, even expressed, or maybe have a different function. The transcriptional regulators might be different in that context. So cellular context is really important.
Even breeds. I mean, just
Yeah.
it's already.
Yeah. I mean,
to we should get a reference genome from every single breed, which is not big, is not bad.
yeah. I
but,
mean, the chicken breeding companies are like, this is great, but can you do it in broilers? Cells from broilers. Like, well, if you can give us cells from broilers, we can do it. But yeah, I mean, they're obviously interested in specific cells from specific chickens. Yeah, I mean, spoken to Andrew Nicky, who's right now busy submitting our grant. And if we can get cells from chickens that prove to be resistant to flu, so for example, you've had a massive outbreak of high path flu, and you tend to get a few chickens that survive. If you can get cells from those chickens, you screen from cells that are resistant in the first place, things like that.
Already getting that, should first have the full annotation of.
Well, yeah.
It's good.
That's not my area.
Yeah. Any other consideration or questions?
I've still got like a few Tunnocks tea cakes if anyone wants them. You can just come and get them. I'll not fling them at you.
Okay, I think for the people online, it will be hard.
That I haven't figured out. Yeah.
Okay, so Juan is asking, is that possible to download the FASTQ file you mentioned?
Sorry, was that again?
Is that possible to download the FASTQ file? Download, yeah.
Yeah, I mean, so that was the issue we were having was I couldn't download it from. I had a bit more time. It was a bit last minute, but I tried to download the FASTQ file from the OneDrive. My postdoc loaded it up onto our OneDrive, downloaded it, and every time I tried to open it, it errored. So in a future workshop, what I hope to have is the unzipped, downloaded FASTA files on the cloud that we could then do that part of it. Oh, sorry.
Answering the person who asks. Yeah. Thank you very much. This was really.
I know, it's surprising when I do something nice, isn't it? Yeah.